Good afternoon, Year 13. Year 12, this one, isn't it? So this is my attempt at topic 4, Key Assessment 1. So let's get on with it. So, first question. First question is a multiple choice question. Which compound found in plants contains nitrogen from nitrate fertilizers? So, nitrogen's the key here. And we know from our work across topics 1 and 2 that we have studied 2 organic molecules that contain nitrogen. Okay, if you look at what we've studied, carbs, lipids, proteins, and nucleic acids. Okay, we know that amino acids contain nitrogen, so that's in protein. Nucleic acids, so the base contains nitrogen. Carbohydrates, as clues the name, carbon, hydrogen and oxygen, so no. And lipids is also a no. So that helps us to then answer this question. If we look at A first, amino acids, yes. DNA, yes. But cellulose is a carbohydrate, so that's a no. Okay. If we look down here, DNA, yes. Enzyme is a protein, so yes. Starch is a carbohydrate, so no. And then here, same thing. Starch is a carbohydrate, so no. Proteins, yes. Cellulose, no. So that leaves us with DNA, yes. Enzymes, yes. As a protein. Amino acids, yes. Our answer here is B. Right, we'll move on now. A bit of maths. Percentage decrease is something that's going to always appear in your exam, so something that you need to get your head around, or sometimes percentage increase or percentage change in general will be in there. So, calculate percentage decrease in nitrate concentrations from the root to the leaf. So, root, leaf. Okay, so we're looking at the decrease. All right, so we're going from the root to the leaf. So, from the root to the leaf, and that gives you an answer. Okay, just no calculator. Right, yeah. 1850, and then we've got to work out the change. Okay, now we're going from the root, so that's going to be at the bottom of our fraction. On top of the fraction, all right, is going to give us our value. So this is the change over the original, all right, times 100, and that should give you a value to the nearest whole number of 88%. Okay, so there you go. Right, describe, keyword here. All right, if you see describe in an exam question, you should dance around exam hall because it is a significantly easier question than anything else. Describe how nitrate ions, so ions is key here, are transported from the root to the leaves. A key way of going wrong here would be to start talking about how the ions are going to get into the root. All right, so that you'd be talking probably about active transport there, but we're from the root. So what we're saying here in this question is those ions are already in the root. So we're talking about how's it getting from the root to the leaves. Okay, here's one of my wonderful diagrams. There's a leaf. So how are we getting from there to there? All right. Now the ions are travelling in the xylem. All right. They're travelling in the xylem, and they're travelling in solution. Okay, they're travelling in the water in solution. So that is all you need to say. Okay, those 2 marking points there is exactly what you're going to need to get. Let's turn over. So, the concentration of magnesium ions in the soil is found to be very low. Explain the effects of the shortage of magnesium ions on the plant. So we've got explain here. Explain means biology. So we need to go a little bit further now, describe the situation going on here. All right. Now, from your work you did yesterday, you'll know that the magnesium is completely to do with chlorophyll. Okay. So And essentially that causes yellow leaves or stunted growth. But, sorry, my clamp stand's getting in the way. This here is a description, really, all right? A description point. So if you're starting with that, that's fine, but you need to then go and explain it. Now, the reason the leaves go yellow is because of a chlorophyll or a lack of chlorophyll, all right? And there is some biology, but we can take that further again as well, all right? So what's the issue with a lack of chlorophyll? Well, the issue with a lack of chlorophyll is a reduced amount of photosynthesis. And the issue with a lack of photosynthesis is you're going to get a reduced production of carbohydrate. All right, and you might want to name a carbohydrate there, particularly for plants. So you might be talking about maybe glucose. You might be talking about something like cellulose. All right, but the key here is that explain. So you might set the scene with that, which actually is a marking point. All right, but to actually get both or all three marking points, you're going to have to do some explanation, and the explanation is about the chlorophyll. And you know that it's more than 2 marks. You're going to have to take that further. And when you're doing explain questions, I would set them up in a way of going A, therefore B, therefore C, and then you might have more therefores going on, but you're just taking that a little bit further and you're showing the examiner your full understanding of that. Okay, and last but not least, we're on to a 6-mark question. All right, devise, lab. Okay, to determine the tolerance category, okay, of tomato plants and lead ions. Now, your teachers will have spoken to you about devise questions and how to answer those. We'll look at those in a moment. It's talking about a lab experiment here. So as soon as you see lab experiment, all right, you need to start thinking about it happening in glass, really, in test tubes. And then this is your actual answer, all right? You want to determine the tolerance category. Now, I think the first thing to do in devise questions is to make sure you're going to get yourself a minimum of 3 or 4 marks out of 6. And to do that, you don't actually have to know a huge amount about what you're doing, okay? And you can follow a very simplified step, which all scientists will do when they're setting up an experiment. And that is as follows. We want to have a look at your independent variable and the range of that. We want to look at the dependent variable and how you're going to measure that. Then we want to look at control variables, okay? And then you should mention repeats, okay? And that's going to give us somewhere in the region of, of our 3 to 4 marks, okay? Now, you're growing plants, so think about what sorts of things you're going to control here, all right? Temp, light intensity, maybe things like concentrate. Let's have just look at the question. Yeah, so that's not mentioned. So the concentrations of the other ions, making sure those are adequate. All right. Perhaps the time. So those sorts of things you're looking at. Your independent variable, your range, you're going to come back to. Your dependent variable, all right, you need to have something that's measurable. Yes? So what you're going to have here is something like, I don't know, height maybe. A number of leaves, perhaps. I've seen before how long it takes to die. It's a bit dodgy, that one. But something you can actually measure, and it's clear that you're actually going to measure that. Okay? Your repeats, when you talk about your repeats, you're always going to calculate a new mean. Again, the range of your independent variable. Now, it gives you a range in the question. It says that we're going from less than 300 to greater than 2,000. Which isn't really a range, and we'll look at that again in a second. So, once you've got your mark scheme, you can see this is level question, all right? Level question is like hopscotch. You've got to get the first bit before you can get to the next box. So early doors in level 1, they're saying they want to see control variables or repeats. And they actually want to see you using tomato plants and lead ions. Lots of people use tomatoes, but you're looking at plants, okay? And the growth of those plants, that's key. In your second box, you need to have your level 1 indicators, all right? But you need to start talking about your range. Now, in this level, they're talking about a range between 300 and 2,000, which is really key because that's given to you in the table, all right? They want a measurable dependent variable, which we've looked at here, but they want a CPAC technique. Now, you're never going to do wonderfully in these questions unless you can actually gauge which CPAC this is to do with. You're never, ever going to be given the exact CPAC, but this clearly is CPAC number 7, which is the mineral ions thing. So we want to see something about plants being grown in test tubes in solutions of lead ions in this case, or whatever ions you're talking about. Okay, so that's going to give us our 4 marks. And then, right, now we need to actually answer the question because we haven't determined the tolerance category of the tomato plants, the lead ions here. Okay, so first things first here, what are we going to do? So let's say we grow it, I don't know, at 1,000 parts per million and it doesn't grow, or it does grow. What does it actually mean? So we need to actually work out, if we're measuring height, for instance, what height equates to the tolerance. Okay, so we need to look at this and go, right, well, you know, we're looking for medium height to be at this height, okay, or even ordering them, or being this number of leaves, or being this colour, or whatever. And we need to go, well, if